Hello, my name is Hannah Galantino-Homer. I'm a veterinary medical scientist and educator, and today I'm going to talk about the flying gallop, witch nails, and mysteries of the horse's hoof. I got a job because of this horse. Barbaro won the Kentucky Derby in 2006 by an impressive 5.5 lengths, but most people remember him for the tragedy that followed when he fractured his right hind leg during the Preakness Stakes. Barbaro's owners sent him to Penn Vet's New Bolton Center. Although he would never run on it again, his fracture was successfully repaired. Sadly, he developed a devastating condition called laminitis in the hoof of the opposite hind limb. Laminitis is a painful and crippling condition resulting from failure of attachment of the hoof to the underlying tissue and bone of the foot. Barbaro received the most advanced care available, including sling support to reduce the weight on his foot and an epidural for pain management. In spite of that care, his front feet also developed laminitis in January 2007, and it was no longer possible to keep him comfortable. Barbaro's owners chose to have him euthanized. They decided to fund a research program at New Bolton Center to better understand laminitis and hopefully to better treat and prevent it. I started that laminitis research program in July 2007. My goal was to use my cell and molecular biology expertise to understand how and why the hoof fails during laminitis. This approach had only recently become possible because an equine genome sequence had just been published. I quickly realized that what we knew about laminitis was limited by a lack of tools for molecular studies in the horse. Looking at it another way, our understanding of laminitis in 2007 was like the 1871 understanding of what horses look like when they gallop. In 1871, artists portrayed horses in this flying gallop pose, floating with the front and hind legs stretched out. If you look at this horse, the anatomical detail is great, but it doesn't resemble what we can see using technology, specifically high-speed photography, to show the actual positions of the limbs during the phase when all four are in the air. Our understanding of laminitis in 2007 was like a flying gallop horse. The anatomy was there, but the molecular studies needed to understand how the hoof works and how it fails were very limited. Edward Muybridge used new technology to understand how horses move. In 1878, he used tripwires and a series of cameras to capture these images to show that horses have their legs tucked up under their bellies during the floating phase of the gallop, not stretched out in the flying gallop pose. New technology dispelled a popular but incorrect understanding of equine locomotion and also led to the development of movies. Like Muybridge's sequential photos, the technological innovation needed to do molecular studies of the equine foot was made possible by the recently published equine genome sequence. The anatomy of the equine foot was well described in 2007. All land mammals, including the mouse and the horse, have nail units. The nail unit includes a nail plate, shown here in this diagram in light blue, which is the claw in the mouse and the hoof wall in the horse. Both have a region shaded dark pink here on the inner surface of the claw or hoof that anchors that nail plate to the underlying distal phalanx bone of the digit, shown in yellow. Unique features of horses are that they have a single digit per foot. The distal phalanx of that digit is fully enclosed within the hoof capsule, and the distal phalanx is actually suspended from the hoof via that specialized inner hoof capsule, so that the hoof wall bears weight instead of a toe and foot pad. How is it possible to suspend a 1,000-pound animal from the inside of a hoof? Unlike animals with nails and claws, animals with hooves increase the surface area of attachment by folding the inner surface of the hoof capsule into lamellae. These folds increase the surface area of the inner hoof capsule and therefore the area that the force of digit suspension is acting on to nearly 1 square meter per horse hoof. The lamellae make the locomotion of a large animal on a single digit mechanically possible. You might have realized that the folds on that hoof capsule diagram couldn't possibly add up to a square meter, and you'd be right. Those primary epidermal lamellae, or PELs, have secondary epidermal lamellae, or SELs, so that if you take a cross-section of the hoof and zoom in, it looks like pine trees with the SELs forming the branches. If you zoom in again, you can see the single layer of basal cells that line the square meter inner surface of the hoof capsule. In 2007, the anatomy of the hoof capsule was very well understood and described down to the SELs, but there was very little information at the molecular level to explain how the individual cells of the SELs maintain their mechanical function and suspend the weight of the horse from the inside of the hoof capsule. Laminitis, shown in the bottom figure, is essentially a failure of the SELs and their connection to the distal phalanx. This results in failure of the suspension of the distal phalanx within the hoof capsule and disruption of the normal anatomy of the foot. But what happens at the cellular level? The microscopy images on the right show basal cells of the SELs expressing a basal cell specific keratin, keratin 14, with a green stain and normal lamellae on top and laminitic lamellae below. Keratins form the tiny internal skeletons of epidermal cells and provide most of the mechanical strength of these cells, but were poorly studied in the hoof. The bottom image from a laminitic horse shows how the distribution of keratin 14 extends into the thickened and distorted lamellar structure. Given the importance of keratins in maintaining the shape and function of these cells, it was shocking how little was known about lamellar keratins. My lab used the newly published equine genome sequence and proteomic analysis to determine that keratin 14 isn't even the major keratin of the lamellae. We discovered a novel keratin, keratin 124, which is twice as abundant as keratin 14. Although there is a version of this keratin in mice, there was almost nothing known about it. My research team developed new tools based on the equine genome sequence to detect lamellar mRNA and proteins. We determined that keratin 124, stained purple in this image, is very precisely restricted to the lamellae and absent from other hoof regions and other epidermal tissues like skin. This seemed to be clear evidence that keratin 124 is important for the normal strength and function of the lamellae. Although the restricted localization of keratin 124 suggests that it has a specialized role, it isn't practical to do the gene knockout or mutation studies in horses that are the gold standard for gene function studies in mice. So there seemed to be no way to prove it. Luckily, a pathologist at Jackson Lab, John Sundberg, had discovered what he called witch nails in mice, and a mutation that knocked out the mouse version of the same keratin. Loss of the mouse keratin disturbs proper structure and function, resulting in misshaped claws that fall off easily. It seems that this specialized keratin has been important in maintaining normal nail unit going back at least 65 million years to a common ancestor, and that role might be especially important in the SELs of single-toed equids. In summary, the equine genome sequence allowed us to discover a specialized keratin restricted to the SELs. That keratin might be essential for maintaining the structure of the lamellae, since the mutation of the mouse version causes witch nails. Finally, SELs and the specialized mechanical properties of unique keratins might have allowed horses to become supreme single-toed athletes like Barbaro, but they also might make it hard to regrow a normal functional hoof after laminitis disrupts the anatomy and keratin localization of the lamellae. Thank you for watching.